Welcome folks. In this video we are going to look at the biology paper 2 for the year 2019. Now we are dealing with a paper that was written by the external candidates. Now here with question 1 from section A, we have figure 1.1 shows red blood cells placed in different solutions. So we can see that there is a solution A. Then we can see after cell A was placed in solution A, after some time we can see the result. The shape of this cell has changed. Then if we look at the solution B, which is cell B, the same happened, but there is a difference in terms of the results. Now before we rush into the finding the answers, first let's look at the process of osmosis. So osmosis is considered as the movement of water molecules from the region of higher concentration, from the region where there is too much water into the region where there is less much water. Now the region where there is less much water is the region that is considered to be highly concentrated. Now here there are 3 solutions that we need to focus into or to put into consideration. The first one, we'll look at the isotonic solution. Now isotonic solution is a type of solution where the concentration of the cell is equal to the concentration of the cell. So if for instance this is our cell here, then we have a solution. Now if we take this solution to have 10% let's say of salt, then the cell will also have 10% of salt. So we can see that we have 2 different regions but of different concentration. Now in this case the diffusion will not take place, and if possible this will show that water will move into the cell, some will move out of the cell, and because of these movements then the size and the shape of the cell will not take place because the concentrations are equal. Now let's say if we want to look at the second one, there is what we call hypertonic solution. Now for hyper, in biology hyper means above. So for hypertonic solution, this is a type of solution where the concentration is much greater than that of a cell. So if for example we have an animal cell here, so we can say that in the solution, if there is 10% of salt solution, then you would find that in a cell there may be 5% of salt solution. So the solution has a much greater concentration than the concentration in a cell. So this means that there is too much water in a cell compared to the solution, because in terms of the concentration, whenever the concentration is high, indicates that the solids are more than the water molecules. So that's what it implies. So here the water will move from the cell into the surrounding. Why? Because the surrounding has a greater concentration, and we are saying water will move from where there is high concentration of water or where there is too much of water. So in a cell there is too much of water, and we can imagine of what will happen. Then what over the third one? The third one is hypotonic solution. Now for hypotonic solution, it is opposite to hypertonic solution. For this one, the cell has a higher concentration. So this means that in the solution there is too much water compared to the water molecules in a cell, and this time water will move from the solution into the cell, and we know what happens. The cell will gain water until at some point the cell will swell and burst. So that is the concept that we are trying to use for us to answer these questions. So in order to summarize, what we are saying is, in a hypertonic solution, the cell loses water. In a hypotonic solution, the cell gains water. That's a key point here. So let's look at the question A. State the type of solution in which each cell was placed. So let's look at cell A. So cell A, we can see the result. So the result, we can see that the cell shrinked. For the cell to shrink, it means that the cell must have lost water molecules into the surrounding. So which solution has the ability of gaining water is the hypertonic solution. So cell A was placed in hypertonic solution. Now we move on to solution B. Solution B, we can see the arrows. The arrows shows that the cell is gaining water, and we are saying the solution that does that is hypotonic solution. So we'll say cell B was placed in hypotonic solution. So the best way to remember, maybe to help you remember these two solutions, remember hyper means above, then hypo means below. So hypo is where the concentration is below, then hyper above. Now let's move on to question B. Give an explanation for your answer in A1 above. So for cell A, explanation, we've seen the arrows. The explanation is giving us or showing us that the cell has lost water into the solution through osmosis. So we can say cell A explanation, the cell lost water into the solution by osmosis, and as a result the cell shrinked. Then the explanation for B, it is just the opposite to this explanation. So we'll say the cell gained water. Gained water from the solution through or by osmosis, and as a result the cell swell and burst. So these were the answers. Now the other advice that I would share with you guys is that the key point that I want you to understand is make sure when you are studying or going through school stuff, just make sure you understand. Then after that, when it comes for you to write an examination or to maybe test whatever, just make sure that you get the concept, you write that concept maybe in your own terminologies. It's hard trying to remember exact word or the way things are written in books. So my point is understand. Then if you understand, get the key points, you can explain in your own languages, as long as you are keeping the main points. Now we will have question 2. Question 2, figure 2.1 shows parts of a rhizopus. So we know what a rhizopus is, or at least we have an idea. So a rhizopus is an example, or it comes from the fungi kingdom. So among the 5 kingdoms that we have under classification, we have this fungi kingdom. Now, I'm sure you may have once encountered or seen a rhizopus, but maybe you didn't know. For example, bread. If bread is kept on a medium where there is high temperature, you'd find that after some times you'll be able to see those greeny stuff like. So those greeny stuff like is what we are referring to as a rhizopus. Now there are, apart from knowing the kingdom where the rhizopus is found, we also have to look at the structures. So the structure from the question 1a, identify the parts labeled C and D. So our C is the sporangium. Then our D, we can see that it's a structure that is holding the sporangium. So this is the sporangiophore. So now these two terms may sometimes will give you like problems when trying to remember which one is this. So you need to find something that should help you to remember which one is the sporangium and which one is the sporangiophore. So you need to find that. Then for structure E, these are stolons. Then the stolons are horizontal. Then the one that are vertical, like this one, these are rhizoid. Now we can now move on to question Roman number 2. Set the function of the part labeled E. So the part labeled E is the stolons. So the function is to secrete a digestive enzyme. So here you can say to secrete, to release, to produce an enzyme. So the key point is just the releasing or production of an enzyme is what the stolons does. Then we have this question B, where we are taught to briefly explain how the organism obtains its food. Now remember, we said these organisms likes to grow on dead matter. So if for example we are looking at a rhizopus that has grown onto a bread, so if this is the bread here, the bread will act as a substrate. And with respect to digestion, by now we need to know what a substrate means. So a substrate in this case is just the substance where the organism is obtaining its nourishment. Now, what is the type of digestion? So the type of digestion that occurs here is extracellular. You see, extracellular types of digestion, why it occurs outside the body of this organism. So the organism, which is the rhizopus, the first one will secrete the enzyme. So we'll secrete the enzyme. Then these enzymes, we are saying they are also considered to be extracellular. So these are extracellular enzymes because they work or found outside of the cell. So they don't work inside of the cells of these rhizopus. They work outside. That's why they are secreted onto the substrate. So whenever these enzymes are secreted on the substrate, they work outside of the body of a rhizopus. Thus the name extracellular enzymes and extracellular types of digestion. So after this complex food substrate is broken into simpler ones, then the simpler ones will now be absorbed by the rhizome. So the function, or one of the functions of the rhizoid, is to now absorb the digested food molecules. So we'll say the rhizopus will secrete an extracellular enzyme. The enzyme will break a more complex substrate into simpler. Then the rhizoid will absorb the simpler molecules. So this is what we can say. So it's just a simplified one, but there is a lot that you are supposed to have in mind. Then so from there, the key point is the first one, there is the secreting of this extracellular enzyme. Then we'll look at the importance of these extracellular enzymes, which is the breaking down of a more complex substrate into simpler one. Then the rhizoid now will come in to do its work to absorb now the simpler one. From there, the organism has the food that it requires. Now we can move on to question C. Rhizopus also produces zygospore. Give one difference between a spore and a zygospore. Now here, we know that if you look at the reproduction under rhizopus can undergo the two types of reproduction. It can undergo asexual reproduction, and we know the asexual reproduction is a reproduction where there is only one parent. Then sexual reproduction, here we require to have two parents, or we require to have the female and the male sex gametes. Now, for these two types of reproduction to occur, it depends on the conditions favoring the rhizopus. So if the conditions favoring asexual, the organism will undergo asexual. If it does the opposite, then we are expected to have sexual reproduction. Now, if you look at these two, a zygospore and a spore, in terms of reproduction, you just have to know that if the organism is undergoing asexual reproduction, it is going to use the spores. If it is undergoing the sexual reproduction, it is going to use the zygospore. So the first difference here we are saying zygospore are used during sexual reproduction. So zygospores are used for sexual reproduction, while spores are used for asexual So that is in terms of the asexual reproduction and the sexual reproduction. Now, what else can we say about this? The other difference that you can consider is in terms of size. So in terms of size, the spores are smaller than the zygospore are larger. That is in terms of spores. Then the other difference is the spores. If you look at the spores are genetically identical to the parent, while zygospores are genetically different. So I've given you at least 3 differences that you need to at least remember. Then we'll move on to question D. Name 2 important roles played by fungi in nature. So the fungi helps the dead organic matter to decay or to decompose or to break. That is our first one. And we can imagine the incident if things were not decaying. By now, I don't know what would have happened. So the space would have been occupied by these, but because of fungi, they help those to decay. As the substances are decaying, we have now space. Then because of the decay, when a substance is decay, like during nitrogen and carbon cycle, some of these nutrients may be recycled. So the other importance is it helps to recycle, helps to recycle the nutrients. Then what else? Then the other use, even if we are taught 2, we can add, since we're just going through this. So the other fungi are used as food. We know that, like if, for example, mushroom, some people, mushroom is considered to be food. Then also fungi can also be used to make medicine. There is that disease of fungal. So in order to treat fungal, we need to use the medicine that was made using fungi. So these are the importances or the roles played by fungi in nature. Not only there are so many, but these are the 4 we can look at for now. Now we move on to question 3. Figure 3.1 shows an incomplete life cycle of a vector. So it's incomplete because we can see that we have 2 missing, denoted by letter G and F. Now, whenever we have a life cycle that consists of 4 stages, we are dealing with a complete metamorphosis. So this is a complete metamorphosis, because under incomplete metamorphosis, there is only there are only 3 stages. Now, here the stages that we need to look at here is, we'll know that the first one, or maybe let's not number these, but we'll just consider, we can consider anyone to be the first, but let's just look at the first one as the adult. So we know that we have the stage for adults, then we have the stage for eggs, larva, and pupa. Now, to some people, it's hard to remember the order of these stages, how these stages are in nature. Sometimes you may remember the 4 stages, but you forget which one comes after this. Now, if there is at least one so that troubles with that, let me show you the key. So the key point is we just have to look at the alphabetical order. So the alphabetical order, we can see that A is the first one in order, then followed by E, then L comes before P, so P is the last one. So this is the order. This is what you can use to identify the order. So adult, eggs, larva, pupa. If you are beginning from eggs, will be eggs, larva, pupa, adult. So this is what should help you. Then we can see that we've answered F and G. So our F, this will be a larva. Then our G, we know that this will be adult. Now, let's look at question A. Name 2 organisms which have a life cycle shown in figure 3.1 above. So the first one is mosquito. Mosquito undergoes this kind of complete metamorphosis. Then the housefly also it has. Then identify the stages labeled F. F, we know it's larva. G, that's adult. Then C, suggest the best habitat where eggs are laid by the named organisms in A above. So what did we say one is? One is mosquito. So mosquito in stagnant water. Stagnant water is just water that is not moving. Stagnant water, or not only stagnant water, also in like tall grasses. Then for organism number 2, which is housefly, this in rotten substances. Rotten substances, like a dumping site. That's where eggs are laid. Then which of the 2 vectors is host to the following pathogens? So between the mosquito and the housefly, which one is the host? So for Plasmodium, we know that a Plasmodium is a pathogen. A pathogen is a microorganism that has the ability to cause infection. Now, Plasmodium lives in a female mosquito, so we'll say Anopheles mosquito. Then for Vibrio cholerae, we have housefly. And sometimes you may encounter a question like, what is the pathogen for cholera? This is the answer, Vibrio cholerae. Then E, explain why it is important to eradicate these 2 vectors. So to eradicate means to remove or to get rid of. So the reason why we are supposed, or people must make sure that they eradicate these, we can say to prevent the spread of diseases. Okay, so now let's move on to question 4. Question 4, figure 4.1 shows a vertebra of a mammal. So we have what we call a vertebral column, and vertebral column, you need to look at the total number of bones that are found there. Now, before we look at that, the total number of bones found in an adult in human being is 206. Then if you look at a child, there is 300. I know some people it will not make sense. How come a small person has a greater number of bones compared to an adult? So the picture here is when a person is growing up, some of these bones will be connecting, therefore reducing in number. So that's a key point here. Now under vertebral column, we have 33 bones in total in number. Now out of these 33, we look at the first one that forms the neck. So the first one that forms the neck are known as the cervical vertebrae. Then in number they are 7. Then which region? They are found on neck region. Sometimes we may encounter a question like name the region where they are found. Then these followed by we have what we call thoracic. Thoracic vertebrae in number 12. Then they are found on the chest region. From there we have lumbar. Five in number. They are found on the abdominal. So abdominal region. Then from there we have sacral. Then in number there are 5 also. Then they are found on the hip region. Then from there we have what we call a coccyx, something like this. In number there are 4. Then they are found on the tail region. So for someone, in case you don't know, a human being has a tail. Now if we add the 7 plus 12 will give us 19. 19 plus 5 will give us 24. 24 plus 5 will give us 29. 29 plus 4 we get our total 33. So these are the types of vertebral bones that are found on the we call it vertebral column, or sometimes the backbone or the spine. So that is what we want to know here. Now the other thing that we need to look at is the way they are arranged like this, and they are 33. Now they only allow a slight movement in terms of the type of joint that we have here is what we call cartilaginous. So it's a cartilaginous type of joint. Remember, there are 3 types of joints that we have under biology. There is the synovial joint. This one allows the free movement. Then cartilaginous, from the word cartilage. This allows a slight movement. Then from there we also have the sutures, where they do not allow any movement. So the point is, under the vertebral column, if you are asked to determine the type of joint that is found here is cartilaginous, and cartilaginous we are saying allows only a small movement. That's why if someone has to bend, will just bend like this. You can't bend like this. It's not possible. Why? In between these vertebrae there is a space. There is a space, and the function of this space is what we call, the function for this space, it is the passage for spinal cord. So this is where the spinal cord passes through. So now if you are able to bend like this, then you can break the spinal cord. So that's the reason why our creator, I'm sure, made it like this, so that you are just allowed a small amount of movement, or maybe that bending you should be in a round manner so that you are not able to break the spinal cord. Now away from there, we can now look at identify the parts of the body, H and I. So our H, this is what we call a central. Then for I, this is a neurospine. So this is a neurospine. In some books you may find it as the spinous process. It's the same. Then B1, in which region of the vertebral column is this vertebrum found? So among these, is it neck region, chest region, abdominal, hip, or tail? So this is found on the neck region. Then give why have we said so. How can we know that this? So if you study these bones carefully, there will be some structures that will help you to differentiate which this from this. Now for this one, we can see the neurospine here. It has this V-shaped here. So whenever you see a V-shaped here, just remember that you are dealing with the cervical that are found on the neck. Then the other is this presence of transverse foramina. Why do we need these spaces? These spaces are the passage for blood cells and the nerves that connects the body to the brain. So this is why they pass through. So we'll say give a reason. It has a transverse. A transverse foramina. Now, C, state the total number of vertebrae in the region of the vertebral column identified. So remember, we said there are 7. Then he said 2 other types of vertebrae found in the vertebral column and their location. We've answered this, so we just have to get, so we'll choose, okay, let's look at this. So we'll say thoracic, then location, chest region, then in number we say it's 12. I'm just adding this point. Then followed by lumbar, and lumbar are the strongest because they carry the heavy body, the upper body. Then they are found on the abdominal. Then number, these are 12. Then we also have the sacral and the coccyx. Now we can move on to question 5. 2 adolescent learners were not sure how the sex of a fetus is determined and wanted to try by engaging in sexual relationships. A1, explain the sex determinants of the fetus. So we just want to look at how do we determine that we are going to have a man or a woman. So the sex determination or the sex determinant for a human being are just the 2 chromosomes, which is the X and Y chromosomes. So these are the 2 important chromosomes that determines the sex. So we'll say a sex of a fetus is determined by 2 sex chromosomes, X and Y. Now what happens if a fetus has 2 X chromosomes? Then we are going to have a female. So a person with 2 X chromosomes is said to be a female, whereas a person with one X and Y, one X and one Y chromosome is said to be a male. So that is the point here. Then for Roman number 2, use a genetic diagram to illustrate your answer. So the genetic diagram for sex determination is like always the same. This doesn't change. So for this one, you can just commit it to memory. So let's say here we have the man. Remember, the man we said must have XY. Then we have a woman here. Here we are going to have XX. Then we'll call this as a genotype of first parental generation. Then what are we going to do? Let's look at random fertilization. So if we look at random fertilization, we separate these, and after separating these, we'll call them as gametes of P1. Then we can do the crossing. So here we have XX. So this is giving us a chance to give us a female, another chance to give us a female here. So you begin with X always, because X is larger than Y. So XY will have the chance for a male, XY the chance for another male. So we can see that we have equal chances. Then these are now genotypes of F1, first filial generation. Now if you look at if the question comes like, what is the percentage for every fertilization to produce a boy? So if we want the chance for a boy, we look at how many? 1, 2. So it's 2 over the total is 4. So the probability is 2 over 4. Then in terms of the chance, we'll calculate as a percentage. We know that this calculation will give us 50%. What of a girl? So for a girl, it's the same. It's 2 over 4 as probability, but multiplied by 100%, this will give us a 50% chance. So this this now is giving us a picture that for every fertilization, if the sperm cell fuses with an ovum, there is a 50% that may result into a female or into a male. So it's that equal. Now we move on to question B. Explain 2 best methods of avoiding pregnancies as adolescents. So 2 best methods. So there are so many. So the best one is abstinence. This is the best one because it is going to give abstinence. So the first, the benefits of abstinence is the person that does this, or the people that does this, are at peace. They have nothing to worry about. Now the second one from this is condomizing. So condomizing comes the second one because this one will prevent not only pregnancies but also the diseases. So that is what we can share for section A. Now Now for section B, we'll do the same way we'll do. We'll just have a discussion. Then from those discussions, you can now create your own paragraphs or notes. Now the first one is, how can we define homeostasis? What do you think homeostasis is? So homeostasis is just considered as the process by which the body makes some changes to make sure that there is maintenance of water, not only water but also some ions. So that is the point under homeostasis. Now under homeostasis we may have osmoregulation process. Now let's look at the first definition for homeostasis. How can we describe? So this homeostasis will say describes how the body regulates its processes to keep the internal conditions as stable as possible. Now the internal conditions, we are looking at the temperature. So water, the amount of water in the body will help us to determine the temperature of a human being. So the key point about homeostasis is all about the temperature, but not only the temperature. We are looking at the water content, the amount of water present, then the ion, which is the salt content. Also we may look at homeostasis also as the blood pH. How is that pH? Is it acidic? Is it neutral? Then apart from blood pH, we have body temperature. Is the temperature above normal, or is it the temperature below normal, or is it normal? Because we know that the average temperature for a human being is 37 degrees Celsius. Above 37, we have what we call hyperthermia. The negative of hyperthermia is that it may cause the enzymes to be denatured, and we know what happens after the enzymes are denatured. The metabolic reactions will slow down, and from that we can say that life will not be there. Then below temperature, which is hypothermia, will cause the enzymes to be inactive. As a result, the digestion and the other life processes will not occur, and the result is death. Then also homeostasis has to do with also the blood glucose. So the point is, if you take homeostasis only with the relations of temperature, no, from now on, what I want you to understand is homeostasis does not only deal with temperature, it deals with it regulates its process to keep the internal conditions. The internal conditions, we are looking at the first one, no? Water content. Then after water content, we may also look at ion. If we look at ion, we are looking at the salt. Then after ion, we may look at this was blood pH. Then from there, body temperature. Then after body temperature, we may have blood glucose. So now let's look at for question B. Briefly explain the role of the following organs in the homeostasis. The first one, the kidney. So the kidney, the first one, in terms of homeostasis, from these internal conditions, which conditions can be maintained or can be helped to be maintained by the kidney? We know that the kidney has to do with the urine formation. So It may help us to maintain water content. So what happens if there is too much water? Then we know that we are going to have a dilute urine. So the kidney will make sure that it produces a dilute urine. Then if we have less, the opposite happens. We are going to have a very concentrated urine. Then also, the kidney will help to maintain the ions, salt ions. Then from there, let's look at the liver. The liver can help to maintain the temperature, because we know that when the temperature reduces, or when the temperature goes below normal, the liver has the function to produce heat. So that regulation of heat from the liver helps to maintain or to raise the body temperature back to its normal. If, for instance, the temperature drops to maybe 34 degrees Celsius, the liver may increase that or may raise that body temperature to normal by producing heat, so that is in terms of temperature. Then it can also help us with the blood glucose. How we look at the storing of glycogen. So these are the points that you can expand in. Then for C, describe when and how the skin reduces loss of heat from the body. Loss of heat. So the first one, we want to describe how. How is this loss of heat produced, and when do you have this? So here we need to look at the first one. We'll say when it's cold. So what happens when it's cold? So the first one, whenever the temperature of the surrounding is low, the body has to make some changes that will make sure that there is a reduction in loss of heat. So when it's cold, the blood vessels will constrict. So to constrict, it means to become smaller in size. Why are these blood vessels constricting? So that there is a reduction in loss of heat. Then we call this one as vasoconstriction. So vasoconstriction. Then the other about the skin, the other thing that we can know about the skin in relation with, if you look at the relation with heat loss, we'll look at what happens to the blood. So the blood vessels, they how can I write this point? So look at the blood vessels will go or move to a deeper position, because whenever it's hot, the blood vessels, they will come on the surface of the skin, so that there is too much loss of heat to reduce the temperature. So the opposite happens when it's cold. So the blood vessels now will move to a deeper position, so they will move away from the skin. So they will constrict by vasoconstriction. Then apart from that, that will move to a deeper point. So here we can now say, when you have if the temperature falls, if the temperature falls, I take that back. If the temperature falls, the cold receptors in skin and hypothalamus communicates by nervous system. That is the first one. Then the coordination center, which is the coordinator, there will work on the heat gain center in hypothalamus. Then the effectors, the arterioles in the skin will will contract, which is vasoconstriction. Then muscles will shiver. There will be that shivering. Then adrenal medulla gland increases the adrenaline, which is secretion of that also. So there is another point of production of that may also help to raise the temperature. So we move on to question 7. Distinguish between open and closed blood circulatory system. So if we look at the open, what is meant by open and closed circulatory system? So if we look at the open circulatory system, it's a kind of circulatory system where the blood does not pass through the blood vessels. So under open, we don't have blood vessels. So under open, blood circulatory system here is different. So now, how is this done? How can we define open? So we can define this as is. We are looking at the system in which the blood is not enclosed in the blood vessels, but is pumped into a cavity called hemocoel. Then examples of organisms that have this kind of circulatory system, we have like crabs. Apart from crabs, also we have spiders. So the key point is here is the blood does not flow through the blood vessels. Wow, for closed blood circulatory system, here the blood passes through the blood vessels. So here we have the blood vessels, and the best example is the human being. So that is the difference. So the difference between these two is the open circulatory system deals with the blood that does not flow through the blood vessels, while under closed blood circulatory system, we are looking at the system in which blood flows inside the body through the vessels, such as we have arteries and veins, e.g. we have animals like human beings. Then for question B, give named examples, explain the terms single and double circulation. So a single circulatory system, for a single circulatory system or circulation, we can see from the word single, it's just the circulation in which blood passes through the body. We can say through the body or the heart once. It passes once. An example of a single, we have fish. Fish has single. Then apart from fish, even amphibians. Then for a double circulation, we are looking at the passing of blood through the heart twice. Now, the best example is human being. We know that if we have the heart, if this is our heart here, then we have the lungs here, then we will consider this one as our body. So there is the movement. There is the movement of blood from the body into the heart, then from the heart into the lungs for gaseous exchange, then from the lungs back to the heart. Then the heart now will pump the blood rich in oxygen to all parts of the body. And if we look at this, we know that we are going to have pulmonary vein, then pulmonary artery. The pulmonary artery transports the deoxygenated blood away from the heart to the lungs. Then the importance of lungs is for gaseous exchange. Then we have the vena cava, which collects all the deoxygenated blood from the body to the heart. Then we have the aorta that supplies now the oxygenated blood. So you can see that there is the first one, the blood passes, Then the second one. So this kind of circulation where blood passes to the heart twice is what we are referring as a double circulation. Then for C, compare and contrast the structure and functions of arteries and veins. So the first one, we can look at the structure. So if maybe I were to draw these two, one has a small lumen, the other one has a large lumen. So a lumen is just the space where the blood passes through. So the first difference is that arteries have small lumen while veins have large lumens. Then the other difference is veins consist of valves. Why? So that they prevent the backflow of blood. Since blood in veins travels at a low pressure, so to prevent that backflow of blood, we need what? The valves. Now the arteries does not contain the valves. Then the other thing, we can see the muscle here. Arteries have thick muscles, then veins have thin muscles. Why do we need thick muscles in the arteries? So that the arteries may carry blood at high pressure. Then what else can we look at here? So we've looked at three differences. The first one, we've looked at the size of the lumen. We're saying artery has a small lumen while veins large. Then if we look at the presence of valves, the arteries does not contain valves while the veins does. Then if we look at the muscle, so if we look at the muscle, the arteries have thick walled muscles while these have thin. Then in terms of transportation, the arteries carry, so that is in terms of functioning. So the arteries carry blood at high pressure while the veins at low pressure. Then the other function is all arteries except one carry oxygenated blood, and the opposite is also true for veins. Then for comparing, the similarity of this is they both consist of the muscles, then they both consist of the lumen. These also they both transport blood. Now we can move on to question 8. Describe sexual feelings. So sexual feelings is just the edge to sexual feelings. We are looking at the edge of wanting to have emotional feelings, emotional satisfaction. That is what we can describe as sexual feelings, like the pleasure for the body. That's what we are considering as this. So this has to do with the pleasure of emotional and physical. So it has to do with the physical. It's where the feelings that satisfy the body, not the soul, the spirit, no, the feelings that only satisfies. It's all about the pleasure of the body. We just want the body to be satisfied. Then explain the causes of our sexual feelings. The first one, age. Age can be the cause. We know that there is a stage of a human being where the sexual feelings are so strong, and there is also a time where the sexual feelings are not strong the way they are. Then apart from age, you can also expand like hormones. Hormones are also responsible. So hormones controls sexual feelings. Then also apart from hormones, we have social interactions. Social interactions, we are looking at relationships. Like for example, it's for example, if we have two individuals, one is in a relationship and the other one is single, the one in a relationship has more chances of engaging into sexual activities. So we have age, then hormones. We can look at the social interactions, which is the relationship. All these can cause sexual feelings. Then step the consequences of acting on sexual feelings without proper planning. So the consequences may be unwanted pregnancies. Remember, there is nothing with a pregnancy. Pregnancy, which gives us a baby or a children's a gift from God. The problem is not the pregnancy. The problem is having sex at a wrong time. That's where the problem is. So unwanted pregnancy. Then the other one is, we may look at this as unwanted diseases. We have such as gonorrhea, we have HIV. Then apart from this, the other consequences of acting on sexual feelings without proper planning, it may ruin somebody's future. So somebody's future may be ruined, because we know that after unwanted pregnancy or disease, their future won't be the same. So it may ruin somebody's future. Even to some people, it may deteriorate thinking capacity. To some people, the thinking may deteriorate. So these are enough points that we can expand at least to get these 6 marks. Then question 9. Give examples in each case. Define the following types of drugs. So the first one, we are looking at therapeutic drugs. So therapeutic drugs, we are just looking at these are just drugs that are used to, the first one, prevent. So we are looking at preventative and reactionary. Thus they bring relief. So these are just drugs that we use to prevent disease. Like there are some of if you look at immunization, most children are given immunization against a certain disease. The immunization is there to prevent. Then also if you look at what apart from immunization, we may look at like coronavirus. So we were given some medicines that we considered to work against the coronavirus. So that is just to prevent. Then for narcotic drugs, these are substances used to treat moderate to severe pain. So these we can say these are just drugs mostly that are taken in order to reduce pain, and under this we may have the common painkillers. Then recreation drugs are drugs that are taken for pleasure. Mostly they are taken for pleasure, like or if it's not for pleasure, like for a mental state, because I'm going through this, let me drink alcohol. So for example, we have alcohol, then we may have cocaine, cannabis, all these are. So to summarize, therapeutic they are just for prevention and reactionary. Then narcotic drugs, they are just for pain relievers. Recreation, these are for pleasure. Then suggest the dangers which may result from abusing the drugs to change body image. So we want to look at the dangers which may result. So to some people, they become extra more nervous. So here we are looking at the nerve impulses. The nerve impulses starts to move at a faster rate compared to the normal one. Then to some people, the opposite may happen. So to some people, the nerve impulses may be slow. So for these people to react, it takes some time to react because their nerve impulses are not functioning properly. Then the abuse of drugs to some may cause diseases. Like if you look at alcohol, too much alcohol is considered to be a slow poison. It may one day give an individual some negatives. Then also smoking. So we may have dangers like causatives of diseases. Then also personal behavior. To some people, you may even see how they are behaving in public, the exposure, dress code changes. So all these can be the negative impacts. Now we can now move on to the last question. Describe the effect of the following on the ecosystem. The first one, agriculture. So what is the effect of agriculture on the ecosystem? So we know that agriculture, we can look at let's look at this one as Stemene system. And we know under Stemene system, there is the cutting of down. So how is that affecting? It may affect soil erosion. So we may have soil erosion. Then apart from soil erosion, we may have global warming, because when they are burning these trees, they produce the carbon dioxide, so which may result in global warming, where we have temperature rising. Then apart from global warming, also agriculture may affect to, like we'll say, habitat for other organisms. For example, organisms such as animals that lives in like forests. The forests act as a habitat, it acts as a shelter. So there is that displacement. These organisms, since they are part of ecosystem and they have roles to play in ecosystem, during agriculture they are disturbed. As a result, the whole ecosystem is disturbed. So we have erosion, we have global warming, which is this is also air pollution. Then we have habitat displacement. This is same as deforestation. So deforestation also has to do with soil erosion. Then it also has to do with land. We say land degradation. So we may have land problem because of this. Then we know that plants give us oxygen during the day. So there will be a disturbance. There will be unstable oxygen in the atmosphere. And you know that we can't survive without this oxygen. Then deforestation also can also have to do with the habitat, which is the area for some of these organisms. Then for B, suggest how the effects described in A above could be minimized. So for agriculture, we have to make sure by we reduce, if possible, we stop chitemene system. Then also we reduce agriculture. Another point here is if we apply too much fertilizer, may cause what we call eutrophication, where there is too much bacteria in water. As a result, the oxygen content of water reduces. So how can we reduce that? So we by reduce depending of fertilizer. So you reduce fertilizer uses. So at least we focus on much on the organic substance, or maybe we teach or we encourage ourselves on crop rotation, where crop rotation is the process whereby you change every year, you change the type of crops that you are going to plant in that particular land. So by, we'll say, encouraging crop rotation. Then for deforestation, here you teach people the importance of afforestation or planting. You plant more plants. Then this will give us the required. So this is what I can share with you guys in this video. Thanks for watching.